Thank you to the Pork Board, because in 2007 when we started this, they were the ones that had trust that we could do this. And I think that's really important, because without their funds to collect the samples, we wouldn't have been able to do all the things that Bob has shown. And I'm going to be looking at mechanisms and trying to probe out what's going on with the infection, and going beyond the GWAS and looking for other alternative things. We couldn't have done what we've done as well without the dedication of the companies that gave us the genetics, so we could use contemporary genetics, so anything we found could be used tomorrow with your breeding pigs. And that's one of the critical things we've done. And this is a review that we wrote on PRRS, and the thing that we take with this is looking at what's the overall thing in the virus load. And as Bob has shown you with the real data, we have some pigs that have early clearance of the virus and others that have slower and others that rebound. And we have the persistent virus. We have data on the tonsils, but I don't have time to talk about it today. And looking at the responses in terms of the antibody, the cells, the interferons, and I'll show you a little bit about that. One of the things that Bob didn't mention is the other genes. You know, we got the first set of genes that Nick showed that 13% of the viral load could be accounted for by the chromosome 4. What about other chromosomes? Well, the next for the viral load is on X, and it's only 1% of that variation. For the weight gain, again, chromosome 4, 9% of the genetic variation. The next is on chromosome 5, 2.6, on chromosome 7, 1.4. So this is like most of the traits that geneticists are looking for. There are multiple genes that are involved. And Bob showed you this, and one of the things that I wanted to point out for the work that I'll be talking about in terms of the regulation of expression is this is day 4, and you're already seeing the effect of chromosome 4. So we talk about the B is the resistance allele, the A is the susceptibility, and what you see is as early as day 4 and day 7, you're beginning to see differences, and by day 11, it's 10 to the minus 16th differences, statistically different. And when we're looking at the gene expression and protein expression, we're focusing on the samples we collected at 4, 7, 11, 14 days. And Bob Rowland was the one who said we should do tempest tubes to collect the RNA, and that was something that turns out to be a huge benefit to our project. And Bob showed you, so we have the susceptible pigs have the higher amount of the virus, and this is on a log scale, so this is 6.5 logs. When you're looking at this, you're a half of a log difference, so it's a major difference in the virus load, and what that translates in to the fact that 14 days you're beginning to see the difference in the weight gain, which translates out to continues to translate out to 42 days when you're looking at the summary of the first 8 trials. And 1 dose of the B-resistant allele was sufficient. So now I'm going to start looking at the transcriptomics, and Bob mentioned that the GBP5, that this is the biological candidate. He talked about the role in innate immunity. It's differentially expressed, and the SNPs show allele-specific expression, and we've done this in 2 trials that I'll show you some of the data in a second trial. It's 100% concordant with the quantitative trait. It's the frame shift causing the earliest stop, and the WUR SNP is associated with the differential splicing, which leads to little or no expression of the wild-type transcript. And there are 12 genes in the region that we saw that, but only 1, the GBP5, showed at multiple time points. So there are multiple other genes in the region are showing association. Only one of them was differentially expressed at all the time points. And another thing we've done is look for copy number variation and not found that. And so with the B allele, what you have is the wild type. You have the wild type expression. With the A allele, what you have is the expression, higher expression of the insert, and so you don't get the proper expression of the gene. Okay, next. Oops, now I go. Okay. So now we're going to be looking at pathways and quantitative traits. If we're looking at the how we did this work, one of the things that we've been pointing out is the large number of pigs that were tested. We have repeated experiments, so it's not a one-off. And that's been really important to prove that this is really important. And it's not one breed. It's not one breeding company. It's the genetics that is there in the commercial pig population. As Bob mentioned, he has 10 freezers. Well, I always try to outdo Bob, so I have 20. But what we have consistently with Maureen Sheehan and Becky Eves, we have consistently shared the experimental samples so that we have redundant samples so we can give them to people and keep the logs of that, and that's been very important. With the tissue samples, we have the blood RNA, the serum, we have the tonsils, but as Bob said, we don't have much in terms of looking at individual cells or the nodes, lymph nodes and things like that, because we tried to do it so we could have large numbers of pigs. Jeff Zimmerman led the big pig experiment, and when I tried to get people to come in on this, people said, oh, those people who do disease studies, 109 pigs, that's the big pig experiment. Well, now we have it that we have 5,000 pigs, and we're really doing so much more that we can get ideas. We have the database that Jim Reese put together, and we keep getting deeper and deeper phenotypes, as Bob pointed out. Disadvantages, we're looking at blood. We don't have the lymph nodes, we don't have the lung tissue, we have mostly no live cell data, and for those of you who've worked with the pig, we don't have the immune reagents and the assays that we might have been able to do had we done in-depth things on the individuals. But the questions I'm going to try to ask are, can we identify indicator gene, protein biomarkers, or pathways for PRRS resistance and susceptibility? And can we use that information to develop alternate PRRS control methods based on this? And the technology. Technology has changed from the time we first started this until now. Could I have a glass of water, please? And we started with microwaves. I won't show you any of that data. And qPCR. We've done a lot of that to verify some of the mechanisms, but we'll be going, thank you. But I'll be showing you the results of RNA-seq data and then the most recent work in our lab using the nanostring encounter. And we wanted to identify candidate gene expression patterns and are there differential with the AB versus AA, and looking for the biomarkers. We took, for trial 3, 16 pigs, looking across, and for PHGC5, more pigs, and looking for not only mRNA but microRNA. I'm only going to be speaking about mRNA data today. Wanted to be sure we had good RNA, made the cDNA libraries, and assess differential genes and use repeated measures, linear model to estimate the effects of the GBP5 marker, the day, and genotype by day interactions. And the first set of data is just looking for what are the patterns of differentially expressed genes. Martin Sharoyan, Tris Togel's group, looked at this and tried to say what are some of the sets of genes that are involved in things. And one of the first things is we have the cluster that is upregulated at the early time point and other genes that are downregulated. And when you look at those, this gets really, sorry, this is too dark. But the most important data is that the cluster 1 is upregulated day 4 and 7. We had 1,200 transcripts, and these are immune response genes, and that's right on target for what we want to be seeing in terms of control of PRRS. With Arun Komandath and Hua Bao and Ixo Choi in my lab and in Graham Plastow's lab, we did more with trial 5, again showing that we have sets of genes that are upregulated day 4 and 7, and when you look at those genes, those are genes involved in programmed cell death, apoptosis, intercellular signaling, proteolysis, and so there are important things that we can look at for genes that are involved in things. And one of the things I keep saying is genes, not a single gene. When I first started this project, I used to say biomarker, but now we know we have to look for sets of genes and pathways involved. And so, but we can involve some genes in GBP5, as we've said. That is, its expression goes up specifically in the AB pigs. They're in the blue. It's up for multiple days, and the blue is lower viremia and better weight gain. But we can also find some other genes in looking for cis genes that overlap for control of viral load or weight gain. And with that, besides GBP5, we can find other genes on chromosome 4 that are candidates for being involved in controlling viral load. And for weight gain, as Bob mentioned, chromosome 7, we have a couple of genes there. And so this type of gene expression studies, so if we look at the CHCR1, which is a regulator of mRNA metabolism, that is higher expressed in the A, B, and BB, but these are alleles on chromosome 7. So these are the alleles of this particular gene that are higher expressed, and those in the red have better weight gain. And so this gives us really good candidates to target for, are there ways that we can control that gene, look for the alleles for that for expression. And another approach that we took in the latest set of data was to look to see if we have PRRS viremia response profiles. So this is looking to say we have in the blue the pigs that don't clear the virus by day 35, 42. In the green, we have pigs that clear it, but they have a much higher level of virus than in the pink, which are the pigs that are fast-acting but low amount of virus. And so using these as categories, can we define mechanisms between the fast and the slow, or between the two fast groups? And so Arun Komandath and Ixo Choi did this work, and we can see that there's differential expression of the SLA on chromosome 7. And one of the nice things about this is that it's higher expressed in the animals that are fast-acting, as well as low viral load. And importantly here is we have differential expression at day 0, 4, 7, 10, 14 at this, so we have higher expression of this, and this may be a marker that we could do without infections, because all of our work has been looking at what happens after infection, and what you prefer to have is markers that you can do without an infection. We also have an unknown gene that has similar characteristics And so that is another potential for a biomarker. When we look, when we compare the fast-acting pigs to the slow-acting, we find another gene, SLC15A, which is involved in uptake and digestion of dietary proteins, as Bob mentioned, the nutrition. And there's no differential expression at day 0, but there is a good differential expression at day 4 and day 7. And so that can be, again, looking at these early time points for the effects. And then another gene that needs to be identified, we see the higher level of expression in the animals that are fast to clear the virus versus those that barely clear the virus. So we have a variety of genes and pathways associated with them to look at. So what have we done with the gene expression studies? We use those to reveal timing and unique control mechanisms. We verified the causative allele. We've looked at pathways that are correlated with controlled PRRS infections. It's clearly important for the early gene expression, 4 to 7 days post-infection. We have some potential targets for quantitative traits, for expression levels of those. And this gives us genes and pathways for a new drug and vaccine targets. So these are clearly where we can look for that next way to try to control. We clearly have the issue of incremental effects of new variation on anti-PRRS response, multiple genes involved, but we know its importance in terms of controlling innate immune pathways. The next thing is looking at protein expression studies. With Jane Christopher Hennings and groups in her lab, we developed the Luminex assay, where you can take a capture antibody for a cytokine and look for the amounts of those levels. With the original work, we did a series of the interleukins and interferons and look for their expression. And we found many of the things, IL-10, there was no difference. The chemokine, CCL2, which is a macrophage-associated chemokine, was very differently expressed in the infected animals. And importantly, interferon alpha at day 4 had a peak expression. In cell culture, interferon alpha is shown to be suppressed. In live pigs, you do have a very high upregulation of interferon alpha expression, and this is done with 3 different trials, samples collected from 3 different trials. So what can we see? The difference in serum biomarkers that we can look for immune protein expression using the bead assays. And what we are doing now is we're continuing efforts to do new sets of monoclonals for IL-6, 13, 17A, interferon beta, interferon beta and interferon beta, interferon gamma. So we have that work is ongoing now, so we can do another set with the new vaccine and challenge trials. We found no significant difference in the serum cytokine levels for resistant AA or BB pigs versus the susceptible AA pigs. So these are things that are involved in other mechanisms for anti-PRRS responses. We know that IL-8, interferon alpha, and CCL2 are significantly altered in low versus high viral load pigs after PRRS infection. And so we have potential serum protein markers that we can use to follow infection responses. And there's a substantial interferon alpha increase immediately after PRRS infections. And so we'll be continuing to explore these biomarkers and cytokines and compare them with the viral replication. And now looking at the newer models with the vaccination and the co-infection challenge studies, looking for pathways. And now rather than full RNA-seq, we're using the 3' RNA-seq data. And this is Jessie Dong has a poster this evening, number 26, and these are some of the subsets from what she's showing. In this model for after vaccination, we have animals that are bled the 0, 4, 7, 11, and 14 after the vaccination, and then they have the challenge of not just PRRS, but PRRS and PCV2B. And so these are 4 groups of 200 commercial crossbred pigs and that were vaccinated with the MLV and then co-infected with PRRS and PCV2. And for the design for looking at the samples, we are comparing the non-vaccinated to the vaccinated and AA or AB, and 7 animals from each of them looking across the infections. And what I'll be showing you is the 3' RNA-seq data that she's been looking at and the nanostring data that we've done on the same RNA samples. And so what she saw at 4 days post-infection, that PRRS vaccine leads to a change in gene expressions predicted to increase viral infection. And what you have is green means that something is downregulated, whereas red is upregulated, and we have a series of genes involved in infection that are downregulated. And with the vaccine, we have a change in gene expression to decrease immune response of cells. And we can see the cyclic, the Fc gamma, and TNF receptors there. And when you look then at day 7, we see that with PRRS vaccinated, these are the responses to the challenge of 7 days post-infection, that the PRRS vaccination resulted in change of gene expression predicted to decrease the inflammatory response, replication of PRRS, and immune response of phagocytes. And so we have the decrease in expression of genes involved with the inflammatory response. We have decrease of genes involved with viral expression and immune response of phagocytes. So with these genes, we can now begin to look at expression and see if we can target specific subsets of genes that are involved in the immune response to PRRS and to the vector of the vaccination on the later response, not just to PRRS, but to PCV2. And so then what we've done recently is looking for using nanostring for selected gene expression. And for those of you who aren't familiar, this is an encounter gene expression assay. It's very sensitive and precise. You can do a direct detection of targets with molecular barcodes without the use of making cDNA. So you do it on RNA specifically. And so rather than the RNA-seq where we're looking at 20,000 genes, we can go for about 200 targets. It's a simple system where there's a reporter, the capture and reporter probes that are designed by nanostring. You have your mRNA, and you make these hybrids, and then you can count for each of the codes how many you have. And you'll see the data that we have for looking for the amounts of the gene expression. If you want to do this, you have to first pick those 230 genes. So a year ago, Linjun Hong in my lab started this by looking at the data from PRRS response studies, from RNA-seq data, and using the Ingenuity pathway to select common gene clusters and pathways. So some of the data that I've just shown you and data from other labs. And then we decided to delete some labs that are repetitive along certain pathways and to select for hub genes. And then we have a final product of 230 genes that we're using for nanostring expression. These are involved in innate and adaptive immune response associated genes, as well as for apoptosis and mitotic pathways that we know are important. And so when we talk about the hub genes, we have a series of gene expressions that come out from hubs, and that's why we targeted those to be more efficient in some of our analyses. And so we have, here's a set of genes that we've done the nanostring expression, and you see for some genes we have genes that can be up to 10,000 copies found, 3,000 copies, and only 400 copies. But all these have the blue is the vaccinated pigs that are AB have a higher level of expression of these genes, including complement 2 and FAS. And so these are ones where we have targets that would be good for looking at expression in the early innate response. And we start with day 4. This is not a day 0 sample. And so we have, this is vaccination at day 0 and challenge at day 28. And for some of them, you can see with MX, where you have the challenge with the PRRS and the PCV2, that you have upregulation for both the AA and the AB that have not been vaccinated. You see the response there that goes up. And so, and with SERPing, what we see is the AA non-vaccinated go up later than the AB that weren't vaccinated when you have the challenge infection. So we have things that we can look for, the early innate response and then the later vaccine response, challenge response. We also have genes that are the same, and you would expect this to have genes that are the same following infection. So Cas1, both the AA and the AB have a similar response, SOX3 and TLR4, whereas TLR7 is differentially expressed with the AA higher than the AB. And when you have, after challenge, both of them are showing upregulation of TLR7. We also, earlier we were looking at the work from Jesse's group on the work on the FC receptors, and we had some early data on granzyme, and we see that there's some upregulation after the challenge, but it's low level with the vaccination. Interferon gamma receptor is higher level expression in the AA than the AB pigs, as well as CD14, which is a macrophage marker. So the animals that have higher viral load have higher macrophage and interferon gamma receptors. So what have we learned from that? We've been able to look at alleles in genomic regions, SNPs and candidate genes, and with the source pig genetics that are correlated with PRRS resistance or susceptibility. We have data on PRRS persistence and didn't have time to show you that. We've looked for the chromosomal regions and the quantitative traits so that we can look for selection procedures to lower the effect of PRRS. We've looked for and uncovered PRRS resistance mechanisms and virus-host interactions. So we hope now that we have these genes that have been identified to help design alternative vaccine or therapeutic approaches. We have this resource of samples that we are willing to share with people and data as well for further analyses. And we are clearly on our way to understanding that we have animals that can be more resistant to PRRS, and we have continued to have our idea of animals with improved resistance, not just to PRRS, but to all respiratory infections. And I just wanted to end that even that was complicated, but that was simple. And if you take just from human vaccine studies, one of the things that you have when you're doing trials, you can do like we have with the RNA-seq, with the proteomics. Metabolomics will be important in order to then look at the next trial and further refine these, and so that we can get the predictive capacity of signatures for independent trials. So as much data as we have, We still need more, but we're getting there so that we can combine this data and give us better predictions. And I want to end with thanks to my collaborators, our funders, and the students, postdocs, and staff have really made this all possible. Thank you.